Hey guys, this is Kendall Terry, and this is part 2 of transport in plants. So this is continued from part 1, and we're looking at number 7 on our PowerPoint, as you can see on the screen. We're looking really, this lecture is going to be focused on the xylem and the phloem of the plant. So how is xylem sap transported? The first you need to think about what is xylem sap, and xylem sap would be water primarily, although there's going to be minerals and different things like that mixed in, and that's why it's called xylem sap as opposed to just saying water in the xylem. So how can it defy gravity is really the question. How does it go up instead of down? Because it should go down. So the first is, of course, cohesion and adhesion. This is nothing new. You've talked about this since middle school. Cohesion, that water's ability to stick to itself. Adhesion, the water's ability to stick to other polar substances. The ability of the water molecule being polar, having a more electronegative oxygen, which creates that polar covalent bond in water. The oxygen being more negative because it's holding on to those electrons a little bit better. The hydrogens being more positive because they're letting go of their electrons more. So then you get this water molecule that has a positive end and a negative end. So what that does is when you get water molecules around each other, they like to adhere to each other, thus adhesion. And they will also stick to other things. And you've seen this with illustrations. This is what makes the paper towel industry so effective, is that water will adhere to the paper towel and it absorbs it or sucks it up. This is also, you can see this inside of your straw. Next time you're at a restaurant and you're drinking out of a straw, if you look where the straw enters the liquid, usually the liquid comes up into the straw a little bit above the surface of the water or the liquid that you're drinking. The reason would be cohesion. So then when we look at xylem sap in the plant, there's a couple different things going on here. One, of course, would be cohesion and adhesion. And you can see that right here. Cohesion and adhesion, the ability of water sticking to itself and to the sides of the cell or the surface of the plant as it travels up the xylem cells. The other is water potential. Now remember, we said water potential was the ability for water to go from high concentration to low concentration. And most of the time we're looking at water potential, we're talking about negative numbers. So it's going to go from the least negative number to the most negative number. And remember, water potential was the combination of both solute potential and pressure potential. So as we go from the soil, which you can see here, the soil is a water potential of negative 0.3 to the root cells of negative 0.6. So it's going from high to low. So water naturally wants to diffuse into the root cells through osmosis, going through the aquaporins that we discussed in class. From the root cells to the trunk xylem or the stem xylem, you get a change from negative 0.6 to about negative 0.8. And this allows you then to go up the tree. So you've got cohesion and adhesion working together, but then you've also got water potential. That water wants to diffuse towards the direction of the most negative number from the stem or tree trunk to the leaf cell walls. It's negative 0.8 to negative 1. From the leaf to the air spaces in the leaf, from the leaf cell walls to the air spaces in the leaf, negative 1 to negative 7. And then finally to the outside area, negative 100. And we talk about that because of transpiration. We've discussed transpiration before. But that process of water going out, because water sticking to itself, also creates that suction in the plant to keep that water flowing up towards the leaves. And thus you end up with adequate supply in the leaf coming from the soil. Transpiration, once again, the loss of water vapor through the leaves. It pulls up from the roots. What controls the loss of water? The stomata, and we'll actually look at the stomata here. So you've got on this side an open stomata, and then over here a closed stomata. I think they're the coolest looking things, especially using the scanning electron microscope that you see here. And I mean, they look like little mouths, open and closed. That's pretty cool looking. What controls the opening and closing of the stomata? This is interesting. It's actually a potassium ion in the guard cells. Remember, the guard cells in this picture would be these cells right here that go around the opening of the stomata. And whether those cells are open or closed is all related to potassium ion in the cell. So now here's a couple pictures, and you can look these up in your book as well. I know the words may not be as visible as I'd like them to be there, so you probably want to look these up some too. But you can see here you've got the guard cells are plump with water. They've got as much water as they can. You've got the cell is turgid, and that causes the stomata to be open. Here the cell is flaccid, meaning it doesn't have as much water as it can hold, and that means that the stomata is closed. So how does this happen? Well, if you look down here at this picture, you've got these red dots. Now the red dots represent potassium ions. So whenever the cell is wanting to open its stomata, it will pump potassium ions into the cell. As the potassium ions are pumped into the cell, this will actually cause water to follow it, because when you pump potassium ions in there, you decrease the solute potential. When you decrease the solute potential, water obviously would go that direction then. So you bring in potassium ions, that brings water with it through diffusion, and that plumps those up. And you can think of this kind of like a balloon animal, if you've ever made balloon animals. If the balloon that you're blowing up is really, really tight, it's hard to get two sections of that balloon to fold over each other. They want to be away from each other, which is exactly what's going on with the guard cells. Whenever the leaf or the plant is wanting to close that stomata, it'll close that stomata by pumping potassium ions out. As the potassium ions are pumped out, water goes with it, because you're decreasing the solute potential then, or technically increasing it depending on how you look at it, because it makes the number less negative. And water goes with it, and then that causes the guard cells to become flaccid, and thus they close. So that explains a lot of the xylem. Xylem deals with both the opening of the stomata because of transpiration and the adhesion, cohesion abilities of water to stick to itself, and of course water potential. There's a lot going on there. You need to go back and look at that again possibly. The next point here is, how is phloem sap transported? Phloem sap is that sugary substance that's made in the leaf. It's stored a lot of times down in the roots. It has to get to the roots for cellular respiration to take place, so there has to be a process of going down, and that process is through phloem cells. So how does phloem load sucrose? Sucrose is the molecule most commonly used to transport sugar throughout the cell, or throughout the plant organism. So here we've got, in this picture, the mesophyll cells, and you can see that picture right there is in the leaf around those air sac areas, makes sucrose, and it will get that sucrose into the phloem cells in one of a couple ways. One will be just through this symplastic route, and we've talked about that route in class. If you don't understand that route, you need to go look that up. The other is actually through an apoplastic route. Now, some plants will choose to use this method of getting sucrose into the phloem cells. Now, I can't tell you the evolutionary reason for that, but some plants use that as a strategy to get that sucrose into the phloem cells. Now, how does it get the sucrose in there? Well, if you remember way back at the beginning of the first lecture on this particular subject, we talked about the co-transporter protein out there that will attach hydrogen ions and sucrose ions or sucrose molecules together, and it will pump them both in. And it uses that hydrogen ion concentration gradient that is developed by this pump here. So that would be the process of the apoplastic route getting into the phloem cells, would be using the hydrogen co-transporter protein. So for that sugar, really, if you just load a cell with sucrose, you can think of really thick syrup or even a molasses. When you think about those, they don't flow very quickly. And so the cell really needs to add something to it to get it to kind of pull down. Because even when you look at like molasses, that's not pure sucrose there. That's still a watered down version. So you need something there to help that have a flowing consistency. So what the plant does is it uses water, which makes sense because when sucrose is in the phloem cell, you've got a very low solute potential. So water wants to diffuse through osmosis from the xylem cells into the phloem cells. And these xylem and phloem cells are pretty close to each other, especially in like the leaf. They're right beside each other. They're right on top of each other. Now, once they get to the stem, they kind of separate a little bit, depending on which type of plant, a eudicot or a monocot. But it's taking water into the phloem cells, and that makes it a little bit more movable, you could say. So you've got a source up here, the leaf. It's loading sucrose. it will also load some water with it. Xylem cells near the phloem will load water in there. So now you've got this watery, sugary mixture that will, through gravity and pressure, flow down towards the roots. Once you get down to the roots, there's some sort of sink cell, and that's a cell that's going to primarily be involved in storing sucrose for the roots, for, say, the respiration to take place, for whatever needs to happen. So you've got some kind of sink cell down here. It's going to start sucking that sucrose out of the phloem. When that happens, it's lowering the water. It's increasing water potential as far as if you think about the number, decreasing the chance for water to stay in the phloem cells, and then water will diffuse back into the xylem cells. So you get a recycling here of water from the xylem to the phloem, from the phloem back to the xylem, and that is all for the purpose of getting that sucrose down to those sink cells, those storage cells that's going to store that sugar until the plant needs it.